Hello and welcome to the immune system, probably part one, potentially of three, because it's quite a big one. And I don't want to spend ages on this. Try to keep these videos short. So we're going to go through the humoral and cell-mediated response and how you write an answer that goes from infection to response. And you can always start your answer by talking about phagocytosis. And you should be able to write, a reasonable amount about this. But just to kind of annotate this diagram, this is a pathogen, general name for something. It could be a virus, it could be a bacterium, and these are its antigens. So likely to be proteins, can be glycoproteins, but not on a virus. And this is our macrophage. You don't need to know macrophage, you need to know phagocyte. So probably put the wrong ones in brackets there. And this is going to carry out phagocytosis. And that's where you need to be able to write, a good answer about lysosomes, vesicle formation, etc. You talk about like fusing phospholipid bilayers, lipid bilayers. Hence, it's in the same topic as the structure of the cell membrane. And some of these phagocytes, they become antigen-presenting cells. So yeah, this is now the antigen that's presented. Okay, and this is going to migrate to lymph nodes. So now we're in the lymph nodes. So this cell here, I'm just going to shade it in. Let's do it red. So this cell here is the same one down here. So this one here is this one. So this is now the same cell. It's just migrated, moved with purpose into the lymph nodes. And you can see that three cells have cottoned onto this one or bound onto it. We've got a B cell on this side. We've got a T helper cell on the top. And this is just one way of thinking about it. Different textbooks will show you this differently. This is just one way of thinking about how they work. Okay, and we're going to talk about the T helper cell first. So this is my T helper cell, and it basically makes a chemical that's going to stimulate the T and B cells. And when I say B cell, TH cell, these are all lymphocytes. And you can say that in an exam, a B cell instead of a B lymphocyte. So this dotted arrow here, this secretes cytokines, which is a chemical stimulant to activate specific B and T cells. Okay, and the reason why they are specific is because the receptors on these cells are complementary to the antigen. So I'm just going to write this over on the blue side for the B cell, but I could say this about any of these cells. So this B cell clone, there are millions of them, has a receptor, and the receptor is an antibody, but the antibody acts as a receptor. So please use the term receptor. This B cell clone has a receptor that is complementary. We have to say how it's complementary. Is it hydrogen bonding? No, it's by shape to this antigen. And crucially, it takes time to find this cell. Yeah, it may take 2 to 3 days to locate this specific B cell. I could say the same for the T cell on the other side as well. So this process of clonal selection, it takes time. If it's the primary immune response, it's going to take 2 to 3 days, in which time you're getting ill. And we'll come back to this, because if you are going through the secondary immune response, this will take place in minutes, hours, and therefore you don't get ill because you've gained immunity. So that, again, that happens on both sides. So yeah, so the T helper cell, or just one last thing about the T helper cell. Let's put a little asterisk there. So my asterisks, yeah. So the T helper cell, or helper T cell, kind of alternates. The T helper cell, yeah, is killed by HIV. So if you've got a HIV infection, your T helper cell numbers drop, and therefore you're less able of doing clonal selection and then activating them. Right, so let's do this side of the diagram first. So this B cell is now this activated B cell. So again, it's the same cell. Yeah, so I'm going to do it in blue again. And whilst I've got my pen going, this one over here is going to be the T cell. And it undergoes clonal expansion. So let's have a look at clonal expansion. This is when I talk about mitosis. So mitosis takes place, and the cells that are produced specialize into plasma cells and memory B cells. See how they have the same receptor type. So there will be other B cells, with different types of receptors. Yeah, all that are complementary to different antigen types. Again, most will be never used. Anyway, let's do our plasma cells first. So this is a specialized B cell. Again, it's still a lymphocyte. These make and secrete antibodies. They're short-lived, as in days, and there are thousands are made. Hundreds of thousands, possibly millions. Okay, so yeah, these make and secrete antibodies, and this is why it's called the humoral response, because the antibodies just become part of the fluid in the body and will just flood the body, and wherever they bump into an antigen, they will bind to it. So that's why it's called the humoral response. Your humors are the fluids in your bodies. Yeah, so that's our humoral response. The memory B cells, these are long-lived. Decades this time, and thousands are made. And these provide active immunity. So yeah, because I've got thousands of them, clonal selection will be a lot faster. So this blue cell basically becomes the same as the other blue cell that is here. So this B cell here, I've just got many copies of it now. So if we re-encounter this spiky antigen again, it won't take 2 to 3 days. It'll happen. All of this will happen faster. I'll have thousands of cells being activated. I won't have one activated B cell. I'll have thousands of activated B cells, and they will do mitosis again. I will refresh my memory cells, and I get my fresh army of plasma cells. Yeah, so that's the humoral response. If you get asked about, how does exposure to a virus cause the body to make antibodies against it, and it's like a 6 marker, then I can talk about phagocytosis for a couple of marks, antigen presenting cell, migrate to the lymph nodes, T helper cells secrete cytokines to activate specific B cells that have a receptor that's complementary to the actual antigen. This then clones by mitosis repeatedly and produces plasma cells that make and secrete antibodies. Done. 6 marks. If a question said, how does exposure to virus cause the cells infected by a virus to be destroyed, I will talk about the cell-mediated side. Now, both these sides happen at the same time. It's just that a question will focus on one side of them. So it's not as if we we do either or here. That's an awful arrow. I'm going to go for a better arrow. Slightly better. So yeah, so this is yeah. At the same time, this is also happening. It's still mitosis here, and I'm still going to get my memory T cells. So again, the same thing happens. It's just subtly different. So, and it's the cytotoxic T cells. These yeah, these locate and kill infected cells. So cells displaying the antigen. Thousands made, and they should be short-lived. Days. So they go and seek out any cell that's got it needs to be a eukaryotic cell. Yeah, so that's that needs to be eukaryotic. They wouldn't be successful at killing a cell with a cell wall like a bacterium because of how they function, and the detail of how they function isn't in the syllabus. They basically like put like a massive like fixed open pore into their cell membrane. It's called perforing, a big like almost like putting a big donut, like putting a pin in a balloon. It just puts an open pore there, and therefore they kind of like burst or because they can't control across their membrane. Memory T cell is the same as it was before. Thousands made, long lived, decades, and they provide immunity. That's active, not passive. And the passive is when you just get like injected with antibodies, or you've got antibodies via the breast milk or via the placenta from your mother. That's passive immunity. This is active immunity. Just zoom out from here. So things to take away. Again, people always think like, how does it know which one to do? A question will ask about the size of this diagram. So, and I didn't say at the end, that this is the cell-mediated response. So if it said describe the cell-mediated response, you talk about, T helper cells again, releasing cytokines that activate specific T cells that have a receptor that's complementary to the specific antigen. They clone by mitosis into cytotoxic T cells and memory T cells, and then the cytotoxic T cells can locate any cell that's displaying the antigen, and they will destroy it. Incidentally, these are the cells that cause like organ rejection. They also cause autoimmune diseases. So these are the cells that destroy the cells in the pancreas for type 1 diabetics. These are also the cells that will attack, like, if you've got intestinal worms and things. So because they're made of eukaryotic cells. So so they'll all got them as well. So so they're often called our natural killer cells. And fun fact, just to finish, all these, the green, the yellow, and the blue are all lymphocytes. And in terms of like the number of them, we have trillions of lymphocytes. And if you had them all in one place, they'd be as big as the liver. So, our liver's like our largest internal organ. The skin's the biggest organ. But, our biggest internal organ is the liver. Well, the lymphocytes would be like a similar size to the liver if they're all in one place. But they're not. They are around our body, knocking around in the lymph, clustering in the lymph nodes, and we have loads and loads of lymph nodes around our body. I'll stop there because I'm pushing 15 minutes. I'm trying to keep these short. Wonderful.